Welcome to Biology Chemistry. This is Unit 1 on the scientific method, lesson number 2, video 3. So we have gone over the first 3 parts of the scientific method. Now we are going to do the last 3 parts in this video. So we have finished part 3, which is design an experiment. So we have a nice controlled experiment. Now we have to perform that experiment. And the key to performing an experiment, other than actually doing what is called for in a procedure, is keeping accurate and detailed records of everything. So that is going to include writing down observations. What did you see? What did you hear or smell during this experiment? You might not use all of those, but you may use some of them. But you're also going to be collecting data. Data are facts, figures, and other evidence that you gather through observation and measurement. And when you are gathering all of this information, all of this data, one of the big keys to success is making sure that it's organized. So organization is really, really important. Making sure that you collect everything, but you also know what you're collecting. You label it properly. You have some kind of spreadsheet, and computers make that so much easier to organize data than before computers were very common. But we can have different displays of data. So we want to be able to share that with a lot of people too. So you want it to be an easy to read format that has a lot of information. So we can have things like charts, graphs, tables, we can have pictures, we could have lists, and we could go on, but those are just some examples of data displays. And we're going to look at this in lesson 3, especially with charts and graphs and tables, in order to know a little bit about how to display some of this data. So that is number 4. Step number 5, now that we have all this data, what do we do with it? So we have to analyze it. So data analysis is the process of interpreting the meaning of the collected data. So not only do we have to gather it, organize it, present it, now we have to do a lot of different things with that. So this is where statistics, which is looking at data and trying to draw conclusions from it, comes into play. And being able to say, well, does this cause this? Does this match up with this? All of those different kinds of questions. So we look for patterns. We look for trends, similarities, relationships, etc., differences, all of that stuff. But the big thing here is you have to keep that goal in mind, which was, did we test the hypothesis? So we always have to keep that in the back of the mind. Does this match up with what we thought it was going to do? So we have to tie it in to the hypothesis. Again, did we prove our hypothesis true, partially true, false, or not tested? Maybe you did all this stuff and you found that one of the variables wasn't controlled and it had an effect. So guess what? You have to go back and redesign your procedure and retest it, run your experiment again, then do your data analysis again. This whole process can take years of somebody studying different things. You also have to look back at your procedure and you look for errors. You might have had a mistake somewhere and you have to go back and fix it. There could be something wrong with the procedure. So there might have been issues with something and you have to go back and correct that. So a lot of times this happens and you have to go back and redesign the experiment. So let's say that you analyze the data and you were able to tie it in with your hypothesis, you controlled for any errors, and you proved your hypothesis was correct. Well, that's not the end of everything. Now you have to communicate the results and you have to come up with some kind of conclusion, which is nothing more than a summary of what was learned and do you support or reject the hypothesis. So if you proved it true, then of course you're going to support your hypothesis. The big thing here is why. So this can take a lot of thinking. Why did I support this? Or why did I reject it? Maybe this had nothing to do. Your hypothesis proved to be false. You have to reject it. But once you do that, then the big thing here, and I can't say this enough, repeat, repeat, repeat. So whenever you do one of these experiments, and let's say that you support the hypothesis, well, it's not the end. Now you have to go back and do the whole thing over again and see if you get the same results. And you might do this 50, 60, 200, 300 times to see if you get the same results every single time. If you do, then you might send it to another group of scientists who will repeat your experiment to see if they get the same results. We do this to verify the hypothesis and results. That's really, really important because we can't say something is 100% in science because there could always be something that comes back and changes that hypothesis a little bit, that changes what we know. We see it all the time. Scientists know something and then they figure out something. Oh, well, this changed that a little bit. So we have a better understanding of it.